Welcome to Chapter 10, The Senses, Part 2. In this PowerPoint presentation, we will focus on the eye and the ear, their structures, functions, and descriptions. Before you view and listen to this PowerPoint presentation, it is suggested that you turn in your PowerPoint packet to page 124, and before we begin, label the eye diagram. You will need to label an eye diagram and be able to give functions of parts on the next quiz or exam. Also, view page 126 as I progress the eye, which is the table of the eye structures and their functions so that you can follow along. The anatomy of the eye. The eye is a hollow fluid-filled sphere. So the eye is going to contain fluid, and when you dissect the eye, often it is very important to use your safety glasses so that the fluid, called aqueous humor, does not squirt out and get on you or in your eye. It is composed of 3 layers or tunics. You'll need to know which structures are located in each layer or tunic. It's divided into chambers. You should have page 124 and 126 of your PowerPoint packet out in front of you to follow along as I describe the eye. The eye is a major component of tests and quizzes. You will need to know their functions, their location, and their description. So we're going to go through each part. The answer key for this will be on Blackboard, but it's better if you can label it before without me and check your answers as we go to make sure that you're understanding key concepts. We're going to go in order with number 1. So again, I'm on page 126 of the PowerPoint packet, which is showing the table which corresponds with the eye structures. So number 1, the first eye structure is the ciliary body. The ciliary body is located here at the top, and it kind of looks like a feathery substance. It is in what's called the vascular tunic or layer, and its job is to hold the lens in place. Our next structure, number 2, is called the suspensory ligament. It is also located in the vascular tunic, and it has the same function as the ciliary body, which is to hold the lens in place. And if you look in the picture, those suspensory ligaments are actually suspending or holding that lens in place. Number 3 is the iris. The iris is the colored part of the eye. It is also located in the vascular tunic. The iris is going to surround the pupil. So the iris could be green or brown or blue, and its job is to regulate the pupil diameter or size. Number 4 is the anterior chamber. Anterior means in front of, and it's in front of the lens. It is behind the cornea. The anterior chamber is also located in the vascular tunic. It is between the cornea and the lens, and it contains something called aqueous humor. Aqueous humor is a liquidy substance, so aqueous anterior, which is that fluid that can go squirting out of the eye during a dissection. The function or role of the anterior chamber in the aqueous humor is to maintain eye pressure, refract light, refract means to bend light, provides nutrients to the inner surface of the eye. Number 5 is the pupil. The pupil is going to be the dark part in the center of your eye. It is also located in the vascular tunic. It regulates the amount of light. So for example, if you're in a dark room, your pupil would dilate and get large to let more light in. If someone were to turn the light on, your pupil would constrict and get smaller to let less light in. Which is why if you go to the eye doctor and they dilate your pupils with eye drops, when you go outside, it's often painful because your pupil wants to constrict to reduce the amount of light, but the drops prevent that from happening. Number 6 is the lens. The lens is an integral part of you being able to see. The lens is basically going to bend all of the light entering the eye and focus it onto the retina, where the action potential will be created. The lens is actually a flexible disc. It looks kind of like a marble, and if you were to dissect a sheep eye, it would be a little bit smaller than a marble, and it does bounce. You could take it out of the sheep eye and bounce it on the table. It is located in the vascular tunic, and it focuses light onto that retina region. Number 7 is the cornea. So the cornea is on the outside. It is in what's called the fibrous tunic. It is a transparent structure that covers the iris and the pupil. Its function is to allow light to enter and focuses light. So light is going to come into the cornea. The cornea is on the outermost region of your eye. If you wear contact lens, then you put your contact lens on top of the cornea, and it's easily scratched or damaged. So sand or any debris or even dirty contacts can scratch your cornea, and it can take several weeks to months for that to heal. Number 8 is the vitreous chamber. The vitreous chamber is going to be behind, or vitreous body, it's the same structure. It is behind the lens. It is in the nervous tunic or layer, and is filled with something called vitreous humor. I suggest that you look at the dissected sheep eye picture in your lab manual. The vitreous humor kind of looks like an egg white, and sitting inside it will be the lens. The function of the vitreous chamber is to hold the lens and retina in place, maintains eye pressure, refracts or blends light, and provides nutrients to the inner surface of the eye. Number 9 is the optic disc. This is a structure that students often have never heard of. The optic disc is also located in the nervous tunic. There are no photoreceptors, which are rods and cones, located there. That is why it's called a blind spot. You can't actually see any images in that optic disc region. This is where the axons exit the eye as the optic nerve. So that's where the eye is then going to become part of the brain, which we'll put together after we get to this presentation on the eye structures. Number 10 is the sclera. The sclera is something you're actually very familiar with. It is the white part of your eye. It is going to be located in the fibrous tunic. It's the firm white outer region. It protects external structures and internal structures, helps maintain eye shape, and provides attachment site for muscles. So what I want us to do before we continue with the next few structures is to kind of look and see where we are. So what you're going to see is right here on the sclera, and this is kind of like peeling an onion. We are on the outside. Again, this is the white part of your eye. Then as we move into the next layer, so we're peeling an onion, we've taken off that first layer, which is the sclera, and we get to the choroid, which is this brown region in this picture. The choroid is the next layer down. Then the third region, the innermost region, is going to be the retina, and this is going to be where the action potential is created. We have 3 layers around your eye. Those 3 layers are also seen right here from inside to outside or outside to inside. So the sclera would be the white part, the choroid would be the brown part, and the yellow in this picture would be the retina. So back to our table, we're going to go to number 11, which is the choroid. That is that brown section in this picture. It is in the vascular tunic. It is the black part of the eye. It contains lots and lots of melanin. You should recall that melanin is a pigment, and what it's going to do is absorb all of those colors or light pigments. The function of the choroid is to deliver oxygen and nutrients to the retina. Number 12 is the retina. The retina is going to actually look like almost a little bit of a thin membrane on the inner surface of your eye. It's in the nervous tunic. It covers the posterior 5/6 of the eye, and it contains photoreceptors, which are rods and cones for light or sight. If your retina becomes detached, you would lose vision because this is where the action potential is going to be created. We'll get into more details of the retina and how the light actually creates action potentials in a few more slides. Last but not least is the optic nerve. You're familiar with the optic nerve from the brain. The optic nerve is not in any of the layers or tunics because it's outside the eyeball itself. It is cranial nerve 2, and this is where the action potential originates. So the eyeball is going to connect to the brain, and we're going to process what we're actually seeing. So this picture, along with the labeling and the functions, is very important for your next quiz or exam. So we're going to move on to some more of the details of the eye. So this is going to show basically all those tunics or layers that we were talking about. So tunic is just a fancy word for layer. So this is just going to go through where each of the layers are located and the structures located in each. So the fibrous tunic is the outermost region or layer. It contains the cornea and the sclera. You will need to know which structures are in which tunic. The vascular tunic is the middle layer, has lots of blood vessels, and it contains the choroid, the ciliary body, suspensory ligaments, the lens, the iris, and the pupil. The nervous tunic is the innermost layer. It includes the retina, something called the pigmented retina, the sensory retina, rods, cones, macula, fovea centralis, and the optic disc. So again, this is basically just taking that same information from the table and putting it into a picture form. So now we're going to look at some specific eye structures in more detail. So right in the center of the screen, you're going to see the lens. Surrounding the lens, you see the ciliary body and the suspensory ligaments. And if you recall, both of those structures' job was to hold the lens in place. And they do by surrounding the lens. Next, we have the iris. The iris's job is to regulate the size of the pupil. The iris has two muscles, circular smooth muscles and radial smooth muscles. So when the pupil is constricted in the first picture, it's going to get smaller and the circular smooth muscles get smaller. In the second picture, the pupil would be dilated, so letting more light in, and the radial smooth muscles would be in control. Retina structures. So in the picture, you're going to see a highlighted kind of an orange-green color that is showing the retina. Again, the retina actually looks like a thin membrane on the inner surface of the eye, but this is where we're actually going to create active potentials and see. The pigmented retina is the outer layer. It keeps light from reflecting back in the eye. The sensory retina contains photoreceptors, rods, and cones. And its job is to detect senses, in this case, light. So in this picture, we're going to see a rod and a cone. These are the photoreceptors. They almost look like antennae. And what you're looking at is basically a modified neuron. So you see the word axon, you see the word synaptic ending. So they're still going to produce an action potential so that whatever you're viewing can actually be seen. So rods are photoreceptors that are sensitive to light. They are 20 times, or you contain 20 times more rods than cones. It can function in dim light, so even if the lights are down, rods are doing their job. Cones are photoreceptors for color vision. So how are you going to remember that? Cones, color, see. There are three types of cones, blue, green, and red. And if you're deficient in any of these cones, you could potentially be colorblind to any of these different color variations. So two other structures, the macula, which is the small spot in the center of the retina. So if you see figure 9.14 at the bottom of the screen, it is showing a picture of the retina. The dark red spot in the center is the macula, and in the center of that is the fovea centralis. So think center of macula, fovea centralis. This is where light is focused when looking directly at an object. So as you're viewing these PowerPoint slides, your fovea centralis is where that image is being directed. The fovea centralis only contains cones. So lots of color, even black and white, which is considered color. Neural pathways for vision. So what you're going to see in the picture is a brain. You're on the underside of the brain, and what you're looking at in this picture is basically an X. The top half of the X, which you've seen in sheet brain dissection, is the optic nerve. That's a portion of the optic nerve. It's actually much longer and would attach to the eyeball. In the middle of the X is the optic chiasm. That's the crossing of the two optic nerves. And then the bottom half of the X is the optic tract. That is where the image is going to be processed deep in the brain. So the optic nerve leaves the eye and exits through optic foramen. Recall a foramen is a hole. So basically the optic nerve is leaving through your orbit in your eyeball, in your skull, and going through that hole. And it's going to attach to the brain. The optic chiasm, also on your brain picture. So these structures that we're going through right now could be in chapter 9, the brain, and they could be in chapter 10, the senses. It's where the two optic nerves connect or cross. Again, that's in the center. The optic tracts are where the image enters the brain's processing center, which we'll look at some pictures of how that works on the next several slides. So in this slide, what you're seeing is basically a top view from above of the brain and the eyeballs. So what you're going to focus on are the two eyes, the fixation point, which is what you're viewing or what you're actually looking at, and that same X appearance that we saw from a different view. So we have your left eye, the optic nerve, the optic chiasm, and the optic tract, and the same on the right side. So what happens is the left eye will see the image, it will cross over, and will actually be viewed or processed in the right hemisphere of the brain. Same with the right eye. So what you're going to notice at the back of this brain picture is that all of those signals are converging in the occipital lobe. The occipital lobe of the brain is in the back, and its job is to process vision, which is why if you have an accident where you hit your head, specifically the back of your head, if you come to or lose consciousness, you often have blurred vision temporarily until that occipital lobe, which controls vision, is healed. So what this picture is showing is basically how your brain and your eye process images. So in the first picture, we have a little picture of a tree, and it is right side up. You can see that in the back of that retina region, which is here way in the back, that tree is flipped upside down. Same with the letter A. So what happens is you quote unquote see the image, your eyeball flips it, and then your brain actually flips it right side up for you to see the image correctly. If someone has dyslexia, the flipping in the brain does not actually happen. So that's why they see images backwards and possibly upside down. So how does the eye work? We're going to put all the steps together. Step 1, the cornea, iris, and pupil let light into the eye. Step 2, the cornea, lens, and humors, aqueous and vitreous, focus light onto the retina. The rods and the cones, which are the photoreceptors in the retina, pick up light and color respectively. Light striking the retina produces an action potential. We call an action potential is a change in charges. We've reached threshold, a stimulus has created this action to happen, and depolarization occurs. This action potential is sent to the occipital lobe of the brain and cranial nerve 2, which is the optic nerve. The brain then processes the image. Eye defects or disorders. So many of us have been to the eye doctor, we wear glasses or contacts, and we're going to talk about why some of those things are occurring. Myopia. Many of you might be what is called nearsighted. If you're nearsighted, you can see things that are close up like reading, but you have a hard time seeing things in a distance like a stop sign. Hence, you're nearsighted. The reason is the image is in front of the retina. In other words, it's falling short of the retina. So here is where we're supposed to focus everything, right here in this retina region, but we're a little bit short. Glasses or contacts, as you can see in letter B, put that image exactly where they're supposed to be on the retina so we can actually see what we're supposed to see. Recall the word hyper means above or beyond. So in hyperopia, you are farsighted. So now you can see things in a distance, but you have difficulty reading things that are close up. Why is that? The image is behind the retina. So again, right here, we're behind the retina. We have the image not focused where we want it to be. Contacts and glasses can, again, correct for that. More eye defects. So there's something called presbyopia. So your lens is very elastic. Again, you could take the lens out of the sheep eye and you could actually bounce it on the table. With most things we've talked about this semester, things that are very elastic as we age become less elastic. So as we get older, we might need to have what are called readers or bifocals that are going to be used for us to see things close up, and that's because we have presbyopia. Astigmatism. Many of you wear glasses or contacts because you have an astigmatism. An astigmatism is an irregular curvature of the lens. So the lens is going to focus that light onto the retina. If the lens is off in little increments or amounts, then the image does not go directly onto the retina region that it's supposed to. So in this picture, you see a candle, and in the first part, the lens is in the correct position and the image is being focused in the correct location. And the second one, you see a candle, and you see that the lens is just off by a little bit, which means the image is not being sent to the retina region it's supposed to. So you see a pinwheel off on the right-hand side. This pinwheel can help you to establish whether or not you have an astigmatism. What you should see if you do not have an astigmatism is 1 through 12, and each line or each number has 3 individual lines. If those lines are blurred and look like one solid line, then you possibly have an astigmatism. Our next one is color blindness. Color blindness is going to result in the absence or deficiency of cones. Remember that cones determine color. Color blindness is more likely or more common in males than in females. It is possible for females to be colorblind, but it is not as common. So over on the side, we have an example to see if you are colorblind. So you should see a number in that circle. And if you see a 74, then you are not colorblind. Glaucoma. So increased pressure in the eyeball. So if someone has something like diabetes, they are more at risk for having things like glaucoma. Their blood pressures are constricting more. So at the eye doctor, they'll take, it's kind of like a pin light and kind of touch it to your eyeball to determine if you have glaucoma. Glaucoma is serious because if you do have glaucoma, then they would want to give you medication so that you don't go blind. Cataracts is a cloudiness of the lens. At the bottom, we see 3 pictures, a healthy eye, a glaucoma eye, and a cataract eye. And basically, a cataract is the formation of this cloudiness due to exposure from UV light. So how could you prevent cataracts? By wearing sunglasses with UV protection. If you do have cataracts, they have surgery that can do that to remove those cataracts. So that is all the information on the eye. We're now going to move to hearing and balance. The outer ear. So just like with the eye, we have 3 functions or 3 sections of the ear. We have the outer ear, which extends from the outside of the head to the eardrum. It contains the auricle and something called the external auditory meatus. We have the middle ear, which is an air-filled chamber. It contains something called the tympanic membrane or the eardrum, the malleus, ecus, and stapes, which are the smallest bones in your ear and your body, and something called the eustachian or auditory tube. Lastly, the inner ear is a fluid-filled chamber that has to do with balance, and it contains the oval window, the vestibule, the semicircular canals, the cochlea, tectorial membrane, and hair cells. You will need to follow along in your PowerPoint packet with the ear diagram in the table, and you will also need to know which of these structures is located in which portion of the ear. So now we're going to go through the structures of the ear. Please make sure page 128 is labeled and follow along to make sure that your labels are correct. Also have page 130 out to make sure that you're following along and going through the structure, location, and functions of each of the parts. So what's interesting about the ear is many of the structures have more than one name, which also makes it confusing. I will have both names on the test or quizzes, or it will be very clear what the correct answer is. So we're going to start with number 1, which is the auricle or pinna, same structure. Depending on the textbook or lab manual, you'll see auricle or pinna. This is what you call your ear. It is the fleshy part on the outside. And if you look as we're going through here, we're going to trace or follow the sound waves through the ear. And at the bottom of this picture, you see kind of a purple region that says outer ear, a blue region that says middle ear, and another green region that says inner ear. And we're going to look at those sections and which structure located where. The auricular pinna is the fleshy part on the outside, and its job is to funnel in sound. Next, we have something called the auditory canal or external auditory meatus. They are the exact same structure. You might know it as your ear canal. So why does it have so many names? Well, auditory for hearing, canal because it's a canal-like passageway. External auditory meatus, same structure. External meaning outside, auditory meaning hearing. A meatus is a canal-like passageway. It is also located in the outer ear. This is the structure in which you would insert your Q-tip. It is the canal that leads to the eardrum, and it also funnels in sound. Next, we have number 3, our tympanic membrane, otherwise known as the eardrum. It is now in the middle ear. It is the separation between the outer and middle ear. It's a very thin membrane that can be easily punctured or damaged. This can happen by loud sounds, having ear pods or earbuds too close to the tympanic membrane for too loud, the same sound over and over like a machinery, or scuba diving and coming up too fast with pressure. You could also puncture the tympanic membrane by putting in an object such as a Q-tip too far into your ear. All of those things would cause a hopefully temporary hearing loss until that tympanic membrane heals. The function of the tympanic membrane is to vibrate and produce sound. Next to the tympanic membrane, we have what are called ossicles. Ossicles is a fancy word for bones. These are the smallest bones in your body, and in order, which you'll need to know, they are the malleus, incus, and stapes. They're also called the hammer, anvil, and stirrup. They are again the smallest bones in your body, and their job is to amplify sound. So all 3 of them together make the sound larger. If these bones were not present, then you would almost sound or hear like you were underwater, like a muffled sound. So you would still hear sounds, but it wouldn't be quite as loud. Number 7 is the oval window. 7 is the oval window. That is going to be the structure in the inner ear that separates the middle ear from the inner ear. Number 8 is the most important structure that we're going to talk about. 8 is the cochlea. If you look at number 8, it kind of looks like a snail shell, and that is its structure. It's a snail shell shaped structure. This is where hearing takes place. Without a cochlea, you would not be able to hear. Sometimes people that are deaf are able to have what's called a cochlear implant, which is putting a part inside their skull to help send those sound vibrations to that cochlear nerve so they can actually hear. We'll be talking more about the cochlea on the next few slides. Number 9 is the semicircular canals. If you look at the picture, they look like half circles. Semi means half. They are filled with fluid. The function of the semicircular canals is to be involved in balance or equilibrium. So think of a child. They like to spin around in a circle, circle, circle. When they stop, they fall down because they're dizzy. So what is really happening there? In the semicircular canals, the fluid is moving. And when the child stops, the fluid continues to move, which makes them dizzy. Number 10 is the vestibule. The vestibule is right between the cochlea and the semicircular canals in the inner ear. It is involved with both hearing and balance. And last but not least is number 11, the auditory or eustachian tube. The auditory eustachian tube is in the middle ear. It is actually going to connect up with your pharynx or your throat. It is going to equalize air pressure between air and the atmospheric pressure. So what that means is as you go up in an airplane or in higher altitudes or scuba diving, your ears may pop, and that's because of this auditory eustachian tube. This is also the location where some children have tubes put in their ears. So when a child is born, this is often has kind of an L-shaped to it, which traps bacteria, which causes ear infections. So they put tubes in, which blocks the bacteria from collecting and therefore reduces ear infections. As we get older, that L shape straightens out, which reduces the amount of bacteria and therefore infections. So that is the ear, and now we are going to put all of that together and talk about hearing and balance. So inside the cochlea, so we're inside that snail shell-shaped structure, we have what's called the tectorial membrane. And this picture's a little bit small, but you can kind of zoom in on it. It is inside the cochlea. It vibrates against something called hair cells. When these hair cells are bent, an action potential is created. For all of these senses, we must have an action potential for us to actually process, in this case, sound. Hair cells are attached to sensory neurons. So again, we're going to have a synapse, a connection between more than one type of neuron. When bent, an action potential is produced, and we're going to hear. So let's put all these together. So please note these are numbered sequence. Step 1. Sound travels in waves through the air and is funneled into the ear by the auricle. That was the fleshy part on the outside. Step 2. Sound then goes to the external auditory or acoustic meatus, same as the auditory canal, to the tympanic membrane, which you know as your eardrum. The tympanic membrane vibrates and sound is amplified by the malleus, incus, and stapes. Those are those small bones in your ear, which transmit the sound to the oval window. Step 4. The oval window sends the vibrations to the cochlea and then the tectorial membrane inside the cochlea. The tectorial membrane vibrates against hair cells. 6. The hair cells become bent and cause an action potential in the temporal lobe of the brain. So in that temporal lobe of the brain, which is on the side of your brain, this is where we're going to process hearing and different sounds and different pitches. The brain then processes that sound. So hearing. So dogs and different animals, they have different ranges of hearing. Humans can hear about a range from 20 to 20,000 hertz. And the most acute, the area that we hear the most is 20,000 to 3,000 hertz. Pitch is the frequency of those sound vibrations. So in other words, how quickly those sound vibrations are being sent, and the higher the pitch, the higher the frequency. Loudness is the intensity of those vibrations. Hearing loss can result in, if you do not wear protective ear wear, whether you're in a factory or in this case you can see a jet engine taking off, so the people on the tarmac, they always have protective headgear. Also, you can see that different decibels, you can see kind of where that range is. So normal conversation is 55 to 65 decibels, a ticking watch is about 20, and then that jet taking off is 140. So any of those on the high end could cause hearing loss. So how is hearing and balance linked? Balance is the same as equilibrium. So because we have fluid in our ear, that also has to do with balance. So static equilibrium is equilibrium where you're not moving. And that is going to be controlled by the structure called the vestibule in our ear. So in this picture, this gentleman is sitting down, static, not moving. And the second picture, dynamic equilibrium, is equilibrium when moving. This would be functioning by the semicircular canals. So their job is to help with dynamic equilibrium. So in this picture, you see the gymnast on the balance beam. So any sport or activity where there's lots of balance involved, this dynamic equilibrium is in charge. So things like gymnastics, figure skating, snowboarding, skateboarding, etc. So that is the end of the information for the senses. Let's see what our study questions look like. Which of the following is not a special sense? Taste, touch, sight, smell. Not a special sense. Letter B, touch. Number 2, the pupil is located in the vascular tunic, fibrous tunic, nervous tunic. A, vascular tunic. Location where hearing takes place, the most specific answer. A, cornea, B, cochlea, C, auricle, D, semicircular canals. Hearing takes place in B, the cochlea. Question 4. The olfactory bulb and tract is cranial nerve 1, 2, 3, or 4? The answer is A, 1. Which of the following is not a taste? Not a taste. Salty, sour, umami, spicy, all are taste. Letter D, spicy, is not a taste.